In previous periods, we discussed about the outbreeding devices, the first type of devices which prevent the self-pollination, that is outbreeding devices. Another topic was that is pollen pistil interaction, the different types of reactions which is occur between the pollen grain and pistil just after the pollination and before the fertilization, that is the pollen pistil interaction. And third topic was the artificial hybridization, which includes emasculation, bagging and dusting. Today the topic is of fertilization. Fertilization, that is fusion of gametes, fusion of fusion of male and female gametes, male and female gametes, that is known as fertilization. Fertilization first time investigated by the Starsberger in lilium and fritillaria plants. Lilium and fritillaria plants. It was first time investigated by the observed by the Starsberger in lilium and fritillaria plants. That is the fertilization means fusion of male gametes and female gametes. That is the fertilization. But in angiosperm and flowering plants, that is double fertilization is there. Double fertilization. Double fertilization, yani here two times of fertilization is there, that is known as double fertilization. One is of first fertilization. In first fertilization, that is it having two steps. That first step is polar nuclease plus another polar nuclei fused together that form secondary nucleus. That is N plus N fertilized together that form 2N. Secondary nucleus. And the second step is of that is secondary nucleus plus first male gamete fertilized together that form triploid nucleus. And this process is known as triple fusion. Triple fusion. And this triploid nucleus will form primary endosperm. Primary endosperm. So this is the first fertilization. Pollen grain means polar nuclei which is moved from polar position, that comes at the center and fertilized together, fused together, that forms secondary nucleus, that N plus N, that is 2N, and the secondary nucleus, that is 2N plus male gamete, and they again combined together that form 3N stage. This is the triple fusion. This process is known as triple fusion, and which form triploid nucleus. Then future, this is also known as primary endosperm. This is the first fertilization. It is also known as vegetative fertilization. Vegetative fertilization, in which three vegetative cells, that is two polar nuclei and third one is of male gametes, which form vegetative tissue also. That's why it is known as vegetative fertilization. Then second fertilization, in which egg cell plus other male gametes that fertilize together to form zygote. It will form embryo. embryo, and this process is known as syngamy. Syngamy. This is known as syngamy, and this is known as triple fusion. So here two times fertilization is there. That's why it is known as double fertilization. It is the unique feature of angiosperm. It is the unique feature of angiosperm. In gymnosperm is also double fertilization is there. In gymnosperm is also double fertilization there. But in gymnosperm, that through the double fertilization, one is primary endosperm is formed, another is of zygote or embryo will formed. So this primary endosperm is developed before fertilization. After fertilization. So that no use of this primary endosperm or endosperm. But here the use of this primary endosperm, because this primary endosperm first developed before the development of embryo. This endosperm develops and provide the nutrition to the developing embryo. So it is the nutritive tissue which provide the nutrition to the developing embryo. But in gymnosperm, it will be developed after the formation of embryo. So it is not utilized in the development of embryo. So wastage of tissue. So only it is uses in the angiosperm, and it is only unique feature of the angiosperm, that is double fertilization. One is of vegetative fertilization, that is triple fusion. Another is of syngamy, in which two gametes, one is the egg cells, female gametes, and male gametes, they fertilize together to form zygote, and the future of zygote that will be formed embryo. The next is of post fertilization events. So three events is there. One is of pre-fertilization of events. Pre-fertilization events, that is that events which is occur before fertilization. That is the formation of gametes and transfer of gametes. That is pre-fertilization units. Then second event is of fertilization, and third event is of post-fertilization events. Post-fertilization events includes development of endosperm, development of endosperm, development of embryo, development of seed, and development of fruit. These are the post-fertilization events which is occur after the fertilization. Development of endosperm, development of embryo, development of seed, and development of fruit. Today we discuss about the development of endosperm. Development of endosperm, that is triploid nucleus. Triploid nucleus which is formed from triple fusion. That is formed from triple fusion, which help in the development of endosperm. That help in the development of endosperm. So development of endosperm is of three types. It is of three types. First is nuclear. In nuclear type of endosperm, firstly this primary endosperm nucleus, which is known as pen, primary endosperm nucleus, it grow and enlarged. It collect the cytoplasm, grow and enlarged. Then after a free nuclear division is there, that's why it form many nuclei. Free nuclear division, it form many nuclei, and The nucleus condition is of 3n. 3n. If we see in root, stem, leaf, gamete, what is the condition, and endosperm? What are the ploidy level of the plants? Ploidy level means how many sets of chromosome is there. That is the ploidy level. How many sets of chromosome is there? That is the ploidy level. Normally, we see that plants is diploid. So, the root is of 2n condition, diploid. Stem is of 2n. Leaf is of 2n. And gametes in which that are formed with the help of meiosis. Gametes are formed with the help of meiosis. So here n condition is there and endosperm having 3n condition. Sometimes questions occur in NEET exam or in different types of competitive exams. If a plant's leaf having or in a plant n equal to 6, if in a plant's n equal to 6, how many chromosomes present in the leaves, how many chromosomes present in the roots, how many chromosomes present in the gametes, and how many chromosomes present in the endosperms. If in a plant's n equal to 6, that time inside the root, that is 2n is equal to 16, 12, and here 2n is equal to 12. Leaf, inside the leaf, 2n is equal to 12. Then inside the gamete, that is n, that is 6, and in endosperm, that is 18 number of chromosomes is there. This is the ploidy level of different part of the plants. Some questions comes in the different competitive exams. So according to ploidy level, the number of chromosomes will be there inside the root, stem, leaves, gametes, or inside the endosperms. So here this is primary endosperm nucleus is of 3n and it is divided with the help of free nuclear division without forming the cell wall. So many nuclei is formed inside the one cell, and then after these nuclei form the cell wall from periphery to center. Cell wall from periphery to center, and it will be become a cellular. It will be become a cellular. This is the free nuclear types of endosperm. In maximum angiosperm, such type of endosperm is there. Maximum angiosperm, such type of endosperm is there, that is of free nuclear. Sometimes the peripheral wall will be cellular and the center portion remain nuclear. Peripheral wall will be cellular and central portion remain the nuclear. It is occur inside the coconut. Coconut. Inside the coconut, both the type of endosperm is there. One is of cellular, which is present towards the periphery, and at the center, the nuclear endosperm is there. This peripheral endosperm, which is cellular, that is known as coconut meat. And the central, which is liquid endosperm is there, that is syncytium. Syncytium means both the type of endosperm present in the coconut, that the coconut meat, which is cellular endosperm, and at the center, that is of syncytium, which is nuclear endosperm. So in maximum angiosperm, this type of endosperm is there, that is of nuclear endosperm. That another type of endosperm, that is of cellular. Cellular endosperm. Inside the cellular endosperm, that is PN, primary endosperm nucleus, enlarged, and it divide with the help of transverse division and to form two cells. Divide with the help of transverse division to form two cells. Then again it divided, means first nuclear division is there after that wall formation is occurred. So after nuclear division, the cell wall is formed. So from beginning, it becomes cellular. Then cell divided by the vertical division, four cell will form. Then after it is continuously divided, and from beginning it is cellular. No free nuclear division is there. So from initial position, the cells divided, firstly nuclear division, then followed by cell wall formation is there, then nuclear division, then cell wall formation is there. So from beginning it will be cellular, that is of cellular type of endosperm. Another type of endosperm, that is of halobial, and it is known as intermediate. Intermediate. Intermediate, it is not nuclear and not cellular. Initial, the primary endosperm nucleus enlarges to collecting cytoplasm. Then after it is divided by the transverse division, it forms two cells. Two cells, one towards the micropyle end, one towards the chalazal end. Then after, in out of these two cells, one is divided by the free nuclear division and another divided by the cellular. In both the cells, both the type of endosperm is there. One is of nuclear, another is of cellular. Means in one cell, it is developed with the help of nuclear endosperm, and another cells developed with the help of cellular endosperms. So then after, it will be again cellular, and after the formation of many nuclei, it becomes cellular. So it is intermediate, means both the types of division is there, nuclear as well as cellular. That's why it is known as intermediate or halobials. Why it is known as halobials? Because it is present in the halobials order of monocot class. Monocots, it is present in the halobials order of monocots. So normally the three types of endosperm is there. One is of nuclear, another is of cellular, and third one is of halobials. These endosperms help in the development of embryo. Means embryo will not develop, which is remain or stayed in the zygote condition. Their development is occur after the development of endosperm. First endosperm will develop, and then after zygote will develop to form embryo, and the embryo, or we can say zygote, receive nutrition from this endosperm. Means zygote receive nutrition from this endosperm, and endosperm receive nutrition from the nucellus. Nucellus, that is the main part of ovule, that is parenchymatous things, and this endosperm receive or develops by the receiving nutrition of nucellus. After the complete development of endosperm, the nucellus will be finished. After the complete development of endosperm, nucellus will be finished. But in some plants, the nucellus will remaining, that is known as perisperm. After the development of endosperm, nucellus will be remaining in the ovule, that is known as perisperm. And perisperm present in the black pepper. Example, that is black pepper. Inside the black pepper, perisperm is there. And after the development of nucellus, it provides nutrition to the developing embryo. So today we discuss these two topics. One is of fertilization. So unique fertilization present in the angiosperm, that is of double fertilization, and then post-fertilization development, in which we studied that development of endosperm, and the three types of endosperm is there, that is nuclear endosperm, cellular endosperm, and halobial endosperm which is present in the angiosperms.